textbooks have always lied to us about what neurons are.
The human brain is the most complex structure in the known universe, and we are in the middle of a scientific revolution to understand its inner workings. Join us for a conversation with world-renowned neuroscientists as they visit Rochester. I am Dr. John Foxe, Director of the Monte Institute for Neuroscience at the University of Rochester, and you are listening to NeURoscience Perspectives.
So Jeffrey Macklis, it's great to have you here in Rochester. I know you've been a repeat visitor to the University of Rochester over the years. So I have a few questions for you about your work and your thoughts about the field and where we're going. And let's kick off with stem cells. When can a person out there with neurological disease, when do you think we'll be at a point where stem cells will be transfected or inserted into the brain and be providing effective therapy?
For the general audience, we'll take this opportunity to do a little bit of education that I think the very term stem cell in the nervous system has been misused to great loss and confusion. Because those of us in the field from the mid-late 90s, when progenitors were identified in the nervous system, used to call them progenitors. And we call them by where they are, of what molecular origin they are, of what neuron types they can become or glial types they can become. And then there's this totally parallel field of embryonic stem cell and iPS stem cell biology. And I think it's only been because of companies and intellectual property and frankly, a lot of bad behaviors in the field where the very term stem cell is thought to be by the general public a thing. Mm-hmm. So I'll declare right now, there is no such thing.
It's not a thing.
It's a range of things.
Right.
It's like, it would be the equivalent of going to a doctor and saying, my friend had his cancer treated with liquid nitrogen. And that would be true on the scalp by the dermatologist of a dermatologic cancer. And then ask if their lung cancer or colon cancer couldn't be treated since it's cancer after all. So I think that imprecision has been purposefully used to mislead. That said, I think there's a huge amount of developmentally guided biology by which many of us in the field are learning the molecular pathways that build individual neuron types or astroglial types or oligodendroglial types of the brain. So I would say for certain human illnesses in which a specific cell type might want to be replaced, and in which a couple of decades of precise developmental knowledge has advanced to the point where one can now try to make that kind of cell type, that one could do that. And that I'd say we already know that hematopoietic stem cells can repopulate the blood system.
Right.
But we have no such thing in the nervous system. Hematopoietic stem cells are those that can make all the lineages and that can engraft in bone marrow. And in the nervous system, we have no such thing as a stem cell that can make all the nervous system lineages. So all of those clinics in Mexico and the Caribbean and other places that I won't mention that promise the therapy that you offered of putting a stem cell into the nervous system to treat something, either are performing, and I'm not going to use this term, but as many of my colleagues term charlatans, or we're talking about those stem cells performing things like immune functions.
Right.
Because there's no way to think of an undifferentiated cell just being placed in the nervous system and knowing what it should become.
When I'm out on my travels around the country and talking to people, particularly we were talking, you and I, about autism spectrum disorder and that, and the number of parents and caregivers who are being drawn into this, who are looking for hope. and being provided very unrealistic answers. And it's important that there's a corrective.
Now I'll give you the optimistic answer. The optimistic answer is within certain human illnesses where pathology of a single cell type at a time is so centrally causal that even partial reversal of that could bring huge, immense impact to humans. I think stem cell biology is going to be exceptionally important. But those illnesses, for example, are going to be very difficult. They're going to range from illnesses where demyelination happens and there's not simply a dysregulation of the myelinating cells already in the brain, because we know there are already progenitors. There are already oligodendroglial progenitor stem cells in the nervous system. So we're going to have to find out if those guys are not operating right. Then one might bring in oligodendroglial progenitors or stem cells that are functioning correctly to be therapeutic. Same thing with astroglia. And in the neuron systems, I think for disorders like spinal cord injury, where it's not replacement of some kind of spinal cord tissue, but rather it's connections from the cerebral cortex to the spinal cord, or from sensory system up, one might imagine very careful cellular engineering to build new developmental embryonic-like neurons that will grow past lesions and provide recovery. That all said, I think what many clinics around the world are doing in the orthopedic, rheumatologic, cerebral palsy, nervous system in certain countries is setting back that progress by decades.
Right. Because of failures to really...
Well, it's not just failure. I think it's no rational basis.
Right.
And by using this confusing term of saying stem cells rather than we are using carefully designed progenitor-derived cells to do X, there's almost like the Wild West of snake oil and cure-all disease potions. You and I are too young to remember that in the 1920s, radiation was claimed to cure all ills.
Right. I'm going to read a quote I read of yours. It'll get us into some of the work that your lab is directly famous for, really. And it's you were talking about neurons. It's like a federal system with both central and independent local government when you're talking about the growth cone. Can you help people understand that distinction between the cell body and its emanations?
The idea is, where the motivating truths are, that textbooks have always lied to us about what neurons are. And they will show a cell in the kidney as my hand with its nucleus and its membrane, and it's that big. And then they'll show a neuron with its nucleus and its membrane, and then I'll say, and that's the axon, and that's its synapse. But that's not true at all. If a neuron had a cell body the size of my hand, its axon would be the size of my pinky in diameter and would extend that same cell on the order of 2 to 3 to 7 to 10 kilometers. So 1.5 to 3 to 5 or 6 miles with precision, and would find a target with accuracy of our two chairs talking to each other.
Right.
And
extraordinary.
That's extraordinary. And it's never been at all clear how the control center nucleus of such a neuron can actually control things that are happening that far away. And it turns out, we know a lot about neurons over the last 30 or 40 years. The fastest signaling method for the tip of this growing pinky 2 miles down the road with precision would be almost like an old stagecoach. And it would take between 8 and 15 hours in each direction to signal back. Mm-hmm. Well, I've gotten to this signpost. 8 hours later, what do I do? And then 8 hours later, turn left. And that doesn't work. So the work that you're alluding to has taken an approach of asking, now that there are extremely powerful methods in molecular biology, to look at every coding RNA or non-coding RNA molecule and every protein, we asked, what is the actual molecular machinery out at those growing tips compared to the same cell? And neurons have long been viewed like that kidney cell. It's one cell, it does a thing. But in fact, what we found is that what we term subcellular compartments have entirely independent control mechanisms, as if they're their own operating cell. They have not just a subset of what's happening back in my hand, but things that you don't even see back in my hand. And I actually liken these things more to NASA designing deep space satellites that are going out past the solar system or into the sun, and they have to go out and be pretty autonomous. They can't radio back. It turns out from Pluto, it takes about 8 to 15 hours for radio signals. It's a remarkable coincidence. So these things appear to be one cell functioning without a single command structure, but rather for decisions based on local context and local needs for decision making, if that makes sense to your question.
I think that was an absolutely elegant explanation. That was really wonderful. Let me switch gears a little
Maybe I can add in,
please,
to connect to you, who's a world expert in autism and intellectual disability and abnormalities of the human intellect developing brain. It turns out that if we think about all of these disabilities, you and I know that there is evidence in different subgroups of humans for not only misrouting circuit abnormalities, but as your work has shown, dysfunction of the actual computations at synapses. Well, the growing ends of those axons, and now I'm going to blow up as if we're going in a big microscope. Now, if my body is the cell body, my axon would be the size of my arm, 6 feet more or less, and 6 inches. And this would then go 20 to 30 to 50 to 70 kilometers. It would take me beyond Canandaigua. I don't know where that would take. Was there
pretty close to Buffalo.
Oh, so close to Buffalo. And that's quite remarkable. But at the end of my growing axon, making all the decisions to turn on the whatever it is, the 490, and then on the this, and then on Route 90, and then it will then transition and develop into synapses. So everything I just told you about, I think of not just about building the circuits, but that means the decisions of whether I'm going to be bolted with steel or with Velcro so I can be plastic and learn to change are all coded in these subcellular organelles of neurons. Mm-hmm. And that means that subtle dysfunction of those mechanisms might account for what goes wrong in the relatively subtle dysfunctions of humans with certain forms of social behavioral autism spectrum disorders. And if that was just a speculation, it would be one thing. But if we look at most of the candidate genes, guess what? Many of them are actually differentially localized with splice variants and various variant forms between cell body and growth cones now that we're looking at these things. So I think this basic cell biology that you asked me about is quite relevant to the immensely complicated human biology of the brain.
Right. I have a colleague in New York, Charlie Schroeder, who always says this term. He says, wiring is computation. And when it comes to the brain, exactly the routing of the wiring systems is an incredible, massively complex system. It's important that things end up where they need to end up.
Right.
We start out life, though, as an infant with massive over-exuberance of neuronal development. Is that part of the process? So that we have, when we talk about pruning, so we lose cells, it's because many of these things maybe there's an imprecision that's in the system that can be tolerated, because as long as most of or substantial number of these neurons end up where they're targeted, so you can function.
Yeah. So I'll give you 2 or 3 answers on that, and I don't mean to be argumentative on this.
Please feel free to be
But you and I grew up being taught that. But I think over the last 20 years, the evidence has become vanishingly small for the 2-to-1 dying out. In fact, one doesn't see that at all during the development of cortex. What one does see, and I think that those data came from the 70s and 80s, where there were fewer and fewer connection axons seen. And there used to, I think, be a textbook view that a neuron sends an axon. But now it's increasingly clear, and my lab was the first to discover these in rodents, but they had been hinted about in non-human primates and humans. There are many neuron types that send from the same nucleus and cell body 2 or 3 distinct axons that precisely target different targets. And then the question you ask, I think, becomes really insightful and important. And it's clear during development that many of them will prune one of these axons. And in fact, some of the human autism-associated genes we have identified are responsible for inaccurate pruning of such axons. So it's another version of your question, but rather than it being, I think it's not just that we have so many extra cells, it can be sloppy. It's that the nervous system has even more elegantly evolved to precisely pull back certain connections. And when those are not pulled back, or when they're pulled back too much, that's, to your friend's point, bad circuitry and bad computation.
Right. Let's take a switch in direction here, because one of the things noticing your extraordinary CV and all the prizes, you're trained as a medical doctor first and foremost, and indeed you kept. Is that correct?
Well, I don't know what first and foremost means. I was trained in science, and I have an MD, and I'm fully trained as a neurologist and as a scientist. I was in a combined medical school, graduate school program at Harvard and MIT. And I expected to have a dual career in the Boston, New York, Baltimore, San Francisco style of 90-10. And I have not seen patients for many years.
And that's my question, right? So I mean, because this is quite a tension, I think, for many. There'll be trainees, MD, PhDs. We train a lot of people with MDs and PhDs who spend a large chunk of their career then really feeling these two things in somewhat in competition. Was that your experience? And when did you make the decision to be completely in basic science?
Well, so I'm going to push back on that. We're doing this in a friendly way, only because I like to be provocative on these things. I don't find binary choices as illustrative as gradations. So my view is that my lab is large enough that some folks are thinking about the questions we were just discussing as the basic building of this amazing processing unit, the brain, its organization, its cellular structure, its molecular development and function. But all of them are also interested in the fact that leads to insights into neurodegenerative disease, ALS in particular, the regeneration of spinal cord injury, autism and intellectual disabilities. So to me, my clinical training, though I took care of patients as a primary, secondary, and tertiary neurologist for, altogether, including residency, about 15 years. I still think about patients centrally. And what I'm struck by is that so many folks see the two as distinct. So I have a new form of translation for them. My form of translation is to say that all the time that I spent with patients understanding human biology, some people who call themselves basic scientists, whatever, I did a second postdoc in the neuropharmacology, drugs, neurobehavior, neuroanatomy, pathobiology, circuit biology of non-human primates. So for those out there, that would mean humans. So I think actually understanding what happens in human disease and knowing which systems and which neurons and which pathologies happen, which don't, actually enable, in many ways, a deeper insight that we've been able to take to attack fundamental problems of molecular and cellular neuroscience. But to me, it was not a switch any more than someone who, quote, was an attorney because they went to law school and were in a firm and are now being a judge. That's not a switch. That's a different form of the same field.
Got it.
And I always wanted to understand, why do humans get these remarkable diseases? And when I first asked in about 1983 as a graduate student, why are these genes being discovered that are mutant in every cell in the body and in every neuron in the brain? And why do only 2 neuron types die in this disease, professor? All the professors would say, and they still do today, we don't have the foggiest idea. And I thought that was a problem.
Yeah, that is
so
a problem.
I set a career goal of defogging that problem, which I think might bring us right back to patients, which is the career goal for the rest of my career.
So a medic at heart, basic science is not, you just don't see them as a distinction there at all.
I think if we go back a century, Santiago Ramon y Cajal, who discovered about 95% of what we know in the nervous system, was originally trained not even as a physician, because that was a higher form, but as a surgeon, sent off to Cuba as a military surgeon, came back retrained as a physician. What does that mean? He wasn't a keen scientist? Right. My mentors at Harvard Medical School, Torsten Wiesel and David Hubel, were trained as physicians. I think their Nobel Prize discovering the understanding of computation in the cerebral cortex, it was not to that deficit. So I actually find the reason I'm giving you such a long answer is I find within our progressive world of science, the siloing and almost xenophobia between the two. Now, to get to your point more pointedly, I don't think it's an easy decision for students deciding whether they should train dually as physician scientists or not. And I think too many don't have that clarity of what that means and end up doing one or the other, but not both.
Right.
And if it hasn't actually benefited the one they do, it's unclear whether the training has been fully worthwhile. But when it does, I don't think it gets measured on the axes that you laid out, if that makes sense. I think some of my very clinically focused hematology-oncology colleagues who are deeply scientifically trained think about cell cycle dynamics and how to disrupt cancers in elegant ways that are not cookie-cutter recipes. And some of my colleagues who are working purely on fly visual system ask questions that will inform what they know from their medical training. about the human visual system. So,
But
I'm a troublemaker.
The field needs troublemakers, people to stir it up and ask the tough questions and call out the nonsense where you see it and issue correctives. And if we can't rely on people like you to do that, we're lost, I think.
The nicer answer to your question is, I think there are ranges of people who work across all kinds of levels. And in many ways, a deeply scientifically trained physician scientist can be somebody who thinks at a level of mammalian, primate, and human biology with keen insights. And that might be very diametrically opposed to somebody who thinks about bacteria or worms or flies. But that's a whole continuum that contributes to how we ultimately think about human disease and its treatment.
Right. You're on the local bus going back into Rochester to get a bit of dinner. And you're talking to somebody who, a conversation strikes up with somebody who doesn't know the scientific enterprise. And they ask you, what on earth, why are my tax dollars being spent to study the life cycle of a fly, of a fruit fly, or the sexual proclivities of a fruit fly? What do you say? How do you explain to somebody how important that is?
When I was growing up, and I'm not sure if you and I grew up in the same place, so I'm not sure if you heard the same jokester, but there was a senator, William Proxmire, who, when I was a tiny child, I don't know why I remember this, I was probably 3 or 4, used to come on television and give what he called the Golden Fleece Award for things like you just described. Why are we being fleeced as taxpayers to study the asexual reproduction of treponemes, worm, or whatever? And it turns out those basic studies of how bacteria replicate, or worms infect, or flies modulate pheromones and sexual attraction have led to countless thousands of human drugs that now have children that used to die of leukemia live, and people who used to die within 2 years with certain neurodegenerative diseases live, and people around the world having antimicrobials that cure hookworms and tapeworms and malaria and countless infections. And it's that we as scientists, the work should be funded, the public should know and want us to do, because it's to the benefit of the public and health. There's a secondary benefit of discovery, but I think that's not really the strongest driving force. Right. But I think it's in some ways the scientific system that forces us to describe things in very technical ways that our tax-paying public doesn't understand. So it's our job to translate for people. So when William Proxmire complained about the asexual replication in helminths, somebody should have said, or Senator Proxmire, how hookworms infect 85% of children in tropical areas from Mississippi and Georgia and Florida down through the central Congo. So it matters to all of us. Yeah. And maybe Proxmire would have said, oh, now that you explained it to me, that's worthwhile.
So Jeffrey, the public have a sort of a notion of scientists beavering away in the lab, maybe not eating, forgetting for a few days to eat. Speak to this business of work-life balance. Now, I happen to know that you were a really excellent squash player before
I was a mediocre competitive squash player.
But how do you keep the balance, the work-life balance?
Well, it depends on whether you ask me or whether you ask my wife and our 2 children. It might be imbalanced, but I think the public sometimes was led astray by television shows and movies of the 60s and 70s, 80s, showing scientists as being some odd form. But
So you don't believe that?
I garden. I move rocks around in the yard. I rake. I ski. I work out every morning. I've played multiple sports, even growing up in Pennsylvania. The quintessential Pennsylvania sports of football and wrestling. So I think scientists can have balance in life.
Right.
I think a more pointed answer is my lab is my second family. We're quite active together. We hike together. We ski together. These kinds of activities often lead to the most informal of brainstorming and of thinking, and are quite useful distractions because scientists don't just go to a lab and pipette. Mm-hmm. It's ultimately a creative discipline, or at least at the highest forms, thinking of new ways to ask questions and to sculpt the discovery of new knowledge.
Students out there working away in labs, in the neuroscience labs, and the cell and molecular biology labs, with their eye on the prize. As somebody who's really had a fantastic career, what's that one pearl of wisdom? What do you say to them? What's going to get them through?
Yeah, so single pearls are always a problem for me.
We have 1 minute. You can do fast.
But I think one might be to identify not a problem right this moment that you want to attack that's important, but what would give your entire career in its inevitable arc meaning and importance, so that if you got there, you would have felt fulfilled. And to some of us, that has some sort of highfalutin kinds of words like changing the world or discovering things that will help large classes. To some people, it might be equally important, but to answer this question that's always been unknown. And if you have that, I think the complexities of life become just steps toward that fascinating goal. And that might be toward human health, toward the betterment of the world, or maybe just discovering the subatomic forces that explain black holes.
Just that.
Just that.
I do think sometimes to describe that moment, and all scientists who've been in the business will know this, where you, for just a brief moment when a discovery's been made in the lab, you and your colleagues, whoever happens to be there at that moment, are the only people on the planet to ever have known this piece of information. It's rather an extraordinary thing. The idea, of course, is to share it afterwards. Thank you so much. That was really fantastic. Thank you very much.